Hello, Yambawa, everyone. We're back. We're back with some more studying. I hope you guys have had a lovely weekend. But now it's time for us to continue with some more studying. Unfortunately, I've procrastinated way too far long. So I'll be starting with some molecular genetics after having an entire week dedicated to neuroanatomy. Now it's back to genetics for the meanwhile. So I think the plan for today will be to just start off and do 3 of them, 3 lectures, and then we should be good to roll with. What is this? We are studying molecular genetics, apparently. Molecular genetics of salivary gland development. So that's going to be the first hour, and then I believe the next hour will be, actually let's check, what is the next hour? I don't have the next one, but we'll be doing that one as the first lecture, and then we'll be studying a couple of others. So that's sort of the game plan for today. Yep. Are we all chilling and billing? All right, should be good. Sounds gross. Yeah, I know, it sounds horrible, ain't it? That's the life that I've signed up for. No gameplay and stuff like that, but this is gonna be just sort of like my regular co-working stream. That's sort of the life that I'm assigned and set up for. That's what it is. But let's go ahead and work and continue on it. I'm going to get it started. Hopefully, I don't know how the lectures are this go around and stuff like that, but hopefully within a few minutes I'll get a gauge of whether or not I can get through with it at like a 1.5 speed or a 1 point speed. But hopefully I can. Hopefully she's like fast enough where I can like, or like slow enough where I can like put it at like 1.5 speed. Anyways, let's where is set access? Ah, there we are. Okay, audio, hello. See if I can hear anything.
Cancer,
but much better.
Okay guys, it's 1 o'clock, so let's start. So my name is Maria Kukuruzinska. I met you when I was doing an orientation for you guys, right? So I'm Associate Dean for Research. I'm also Interim Chair of the Department of Translational and Dental Medicine. And I do research, and I've actually done research on salivary gland development, so this is really sort of my neck of the woods. Most recently, I work mostly on head and neck cancer, but I will show you some of the cool pictures and images of salivary glands, and also some of the regulatory mechanisms that we now know are involved in driving the formation of mature salivary glands, and also how defects in certain components that are absolutely critical drivers of salivary gland development, how these defects manifest themselves as mutations, and they impair, generate morbidity basically in individuals' lives. So,
all right, I think she's fat. I think she's slow enough where I can put this at like 1.5.
Here we go.
There we go.
All right, it's better to do manually. So first, what I did is I organized this lecture for you so that it's going to be easy to digest. And actually, there's quite a lot of text in there. But the reason why I put text is so that you can read through and understand the material better. If I just put images, I think sometimes it's very difficult to make connections. So some of the information here is a little bit redundant, but I think it's going to be able to, by reiterating some of the points, I think it's going to be easier for you to understand it. So, as you probably know, salivary glands look like grapes.
How are
and
you?
The image you see on top there
How's classes going for you?
Basically an image of a salivary gland, but this is from a mouse model, and I will explain why we use mouse models, because clearly we cannot get salivary glands from human and follow the development. So we use mice as a model system. But basically, what do we know about the structure? They are tubular acinar exocrine organs. Exocrine meaning that they secrete, right? So they secrete saliva, but saliva is secreted by multiple cell types, and they have different functions in driving composition of saliva. And so they typically generate, saliva is typically generated by 3 types
You're
of
waiting
glands.
for your ride right now?
There's a
Are you done with school?
There's a sublingual gland and submandibular gland. Collectively, these three major pairs of
Way to
glands
survive in there,
contribute about
Chief.
90% of saliva. The rest comes from minor salivary glands, something like, as you probably know, you have in the inner part of your lip, you have salivary glands. Salivary glands are also a part of your tongue.
Carotid,
And these are about
sublingual.
600 to 1,000 minor salivary glands, and they
90%.
contribute the rest of its saliva to the overall composition that you are
I do not like
experiencing in your
how
oral cavity. So
she used
parotid
white
gland
text.
produces mostly serous
Mixed-
acini, is composed of serous acini,
I
and
can't
they mostly
highlight
produce the proteinaceous
with
compounds.
the yellow
So a lot
marker.
of proteins that are found in saliva are secreted by parotid gland. Sublingual gland, on the other hand,
Oh my
consists
God,
of
such a
mostly
terrible
mucus-like
color
cells,
choice she
and
chose.
they deliver mucins to
White
the
on
saliva.
black.
And then submandibular gland actually is a gland that we study most readily because this gland is easy to dissect and it's very easy to follow developmentally. And this gland consists of, or is composed of two types of cells, and they're mixed cells, both the cells that produce proteins and also cells that produce mucins.
Hello,
so these are
hit.
called mucoacinar cells. The important, and probably you know this, that production of saliva is regulated very much by the nervous system. And as you probably know, that sometimes when you, let's say, eat something which is sour, you secrete more saliva. If you get stressed out, you may have differences in your salivary flow. At night, your saliva flow is much reduced versus during the daytime. And this is all controlled by the nervous system. And it is now known that the parasympathetic innervation and sympathetic branch of the nervous system are very much involved in regulating what kind of secretions you get in saliva. And so parasympathetic branch regulates mostly secretion of water. And this is through water channels. Some of the typical water channels have been discovered in saliva. And salivary glands include aquaporin 5. And actually, Peter Argy at Johns Hopkins ended up getting a Nobel Prize by studies of these water channels.
Oh, there you go.
Sympathetic branch secretes digestive. Sympathetic branch regulates the secretion of digestive enzymes such as amylase.
I wonder if we have to know these
amylase is
receptors.
one of
I
the
hope
key
not.
enzymes in your saliva. And what it does, it basically drives the digestion of carbohydrates. So like if you eat a lot of starch, bread, rice, pasta, all of these things are actually digested by the fact that you have amylase being produced in your saliva. So salivary secretions are generated by end buds, and these are the acinar units, and they consist of acinar cells that have sidedness, and we will discuss it when I show you the structure. And these cells secrete into the intercalated ducts, and then there's a whole system of ducts that allows saliva to flow from these secretory cells, secretory units, through the whole channels, networks of ducts, all the way to the major excretory duct, which is emptying into the oral cavity. So these include intercalated ducts, striated ducts, and major excretory duct. So major functions of saliva, you probably know this too, is digestion, it's being able to speak clearly, it's taste, and remineralization. And this relates to your teeth and the fact that saliva is absolutely critical for the maintenance of teeth. And if you, I'm not sure if you're familiar from your studies, but let's say individuals who are on drugs that are impairing salivary flow, and these usually have to do with mental disorders, these individuals end up losing teeth because they do not produce proper amounts of saliva. So in addition, we need to have a balance in the oral microbiome. And as you probably know, microbiome is extremely important towards maintaining a very healthy oral health, and that impacts our systemic health. So oral microbiome modulates also the way the host of the cells in your oral cavity respond to bacteria and respond to fungal infections, respond to other aspects of maybe toxic substances. In addition, it provides also immune components, and especially innate immunity, because we have a lot of immunoglobulins in our saliva also. And these can drive not just the innate immunity per se, but also activation of adaptive immunity, or acquired immunity. So salivary glands, as you see here, in these, so these are sort of these wonderful looking grape-like structures. They are consisting of many, many buds. And these buds with tubuloacinar extensions are very critical for producing these massive amounts of proteins and mucins and water that gets transported through the ducts. And why do we have so many strange aspects of it? Why does it look like a grape? Because salivary glands are confined to your jaw, to your craniofacial region. And in order to be able to produce the amount of saliva that you need for daily living, you need to have a huge amount of surface area, right? And this huge amount of surface area is generated by having so many different components that collectively will give you the surface area. So typically what we do is we generate the salivary gland through the process of branching morphogenesis. And this branching morphogenesis generates these multiple buds and multiple networks of ducts that allow you then to produce the amount of saliva that is required for you to be comfortable in your daily living. So this is sort of an example of branching morphogenesis. Where
She's going so
basically
fast.
What we do is we drive formation of these
even like
very
at
complex
normal
structure,
speed.
and this is from
This
a
is
mouse
just
model
really too
from a
fast.
single bud, which then undergoes multiple morphogenetic changes that ultimately become this very complex structure
It's
that
so
looks
cold,
like a great
dude. It was
bunch of
so
grapes.
cold over here too, dude.
And
it was
this involves
windy.
rearrangement of a single
I
epithelial
feel
bud
that
through
I
multiple
miss already
processes, and we'll
like
discuss them, but involve
I miss
clefting of
fall
the bud.
weather. We're
You
getting
see this,
into
actually,
winter
these
weather.
different buds are generated from this bud because it undergoes clefting and branching and extension of sizes of these buds that ultimately undergo repetitive types of clefting, and you end up with this enormous structure that is a mature salivary gland.
Immature salivary
Much
gland
of the
over
knowledge
here.
today that we have comes from studies of mouse model, as I said, because we can actually dissect
Oh, I
glands from
just
a single bud
realized.
stage and then allow them to grow ex vivo
Are you ready for
in
this, bro?
external conditions. And these will then undergo this process of branching morphogenesis when supplied with appropriate nutrients and will generate this particular structure. So this is just an example of mouse models. As you probably know, we can manipulate mouse, embryonic mouse in very many different ways. We can genetically engineer mice now. And this is just an example showing you that here, for instance, is a keratin 14, which is a protein that's expressed specifically by epithelia. Either
Should I slow it down?
Either in the oral epithelium, in the salivary glands, or also in the skin. And
I'm not gonna lie to you.
That this green fluorescence protein that's attached to this particular promoter of that protein is going to light up a lot of the surface epithelium, including salivary glands. So knowing this, we can trace single cells from the bud stage all the way to the process of generating this mature gland in a very simple way. This actually, so this is an in vivo system. This is an ex vivo system where we dissect these buds and we grow them for 12 hours.
I'm talking to you guys in general.
And that actually will allow you to show that during that particular time period,
actually
you are
listening
getting
on
additional
this.
branching. You are getting actually all of this formation of new buds and clefting and the processes that are involved in generating more developmentally advanced gland. So this is just to show you that in fact
your teacher's speed running the listen. Yeah, no, I like put her at 1.5 speed. I think it's like way too fast now.
Mammary gland, lungs,
I'm starting to
kidneys,
regret doing that.
Prostate, in fact, also tooth and hair. All of these structures develop, all of these organs develop through branching morphogenesis.
She's actually speaking fast.
So salivary gland, in some ways, has been in the forefront of understanding the process of branching morphogenesis because we can dissect it out very easily.
She's talking so fast, I know.
And we can grow it and we can experimentally perturb it and then look at the consequences of what happens to the outcome. So this is sort of the branching morphogenesis in a nutshell, where you see the different processes, how it starts and how it proceeds towards getting a differentiated functional gland.
I don't even know what's the important stuff to know.
Typically what happens, and this is from a mouse model, we see that there is initially condensation of a mesenchyme, and the epithelial placode, which is just surrounding this condensed mesenchyme, is going to invaginate into the mesenchyme. And this occurs at embryonic day about 11 and a half in the mouse. And in human, it happens at 6 to 8 weeks of intrauterine life. Then there's a process of crosstalk between this bud that will be growing. Bud condenses, these cells condense to form a bud, and the bud is going to be growing into the underlying mesenchyme. And there's a crosstalk in a lot of genetic and growth factor and bone morphogenetic proteins that goes back and forth, sort of like in the tooth development, that basically generates further expansion of this bud. At embryonic day 13, the primary stalk starts getting elongated, and this is where the major excretory duct is going to be formed, because as you can tell, it's going to be opening to the exterior and it's to the oral cavity. And this duct elongation is going to be occurring, but it also will involve parasympathetic innervation already. And as you see, we see that there are these buds that are forming, and they are generated because now you have a process of clefting. And this clefting is, again, highly orchestrated and requires particular regulatory molecules that are coming both from the outside extrinsic and intrinsic factors. And then branching is just expanding through
Oh my God, that's not in the
days
notes.
of embryonic development. And there is more the parasympathetic innervation. The green is the nerves, neurons. And ultimately at embryonic day 14, you see that you already have these major structures that involve branched aspects of the acinar cells, also the ductal structures that appear to be now getting luminized. And all of this together is still just a mature gland that is continually developing until about embryonic day 18 and a half, at which point the animals are born. So this is very much dependent.
This is an example of a mice, right?
On so-called progenitor
that makes
cells.
Sense.
And the progenitor cells involve combination of stem cells that are undergoing some lineage determination already. And as you know, stem cells are very important. There is a lot of controversy about using human stem cells, but what we do know is that adults, for instance, have stem cells, and these adult stem cells can be guided to generate either tissue repair, and there's a huge interest currently in that, or they can actually be either in some ways inhibited if you have inappropriate growth, like for instance in cancer, and we'll talk about it very briefly at the end. So what happens, this is a duct, and what happens here is that we see progenitor cells in so-called K5, keratin 5 expressing cells, and that's sort of in the outer region by the where you see SOX2. And SOX2 is a transcription factor that drives stem cell behavior. And it's required for stem cells to be maintained and to be expanded. Keratin 19 marked cells in the luminal part, and these keratin 19 cells are still immature, but they're not quite stem cells. They're already advanced more along the lineage of development.
Hope you're having a good day.
So
did you look at the study guide? I am not looked at it.
Our colleague who
I feel like I should be.
That there is a different level of composition.
You should, what's it called? Tell me if something is on the study guide.
Salivary gland, this is a mandibular gland.
I maybe should put it up on the side.
19 cells,
There's just too many windows.
Maybe what, 25% of the entire cell population. Then there are
I'm good. How are you? I'm dying.
Positive cells,
because of school.
5 cells, and there are keratin 5
but I'm okay.
19 negative cells.
I could be napping right now, but I'm studying.
Is that you always have a collection of heterogeneous cells. Not all cells are the same. They're always changing. They're changing their identities. And this is perhaps one of the greater challenges when we deal with diseases, because in disease states, you have this exactly the same thing happening. You have de-differentiation, you have loss of identities, or changes in cell identities. We call it cell plasticity, which right now is the hallmark of many diseases.
I am not following
so,
this.
This is actually something that was done in my lab. And what you see is the single bud, which is like just a bud that is actually, we stained it with beta-catenin, which is one of the components of Wnt signaling, but it also is a member of catenin junctions or adhesion molecules. So beta-catenin sort of stains the entire epithelium. And this is what happens when you generate the full gland. And now you have in red, you have ductal structures defined. And the red here is actually defined by filamentous actin, because filamentous actin defines a lot of organs that are lined with sort of actinomycin structures, so that these ducts can actually either contract or also deliver particular components to the oral cavity. So you see these ducts are sort of expanding and going into different stages of these different asana structures. At the very end, you have the most differentiated structures here. And what we do know is if you start looking already at the bud stage, you see at the very edge, this is the outer part of the bud that is in contact with the basement membrane.
I'm going to need practice questions on these.
You already have cells that appear to have shape. And these, once cells attain shape, they are more towards polarized behavior. In other words, this needs to be occurring when you have a differentiated phenotype and functional differentiation. We see that these cells are already present at a single bud stage. So what it tells you is that there's already lineage commitment of some cells very early, at the early stage of bud, just when bud forms. And below, I show you a sort of a cartoon that is telling you what exactly happens. So these are the cells that are being maintained. They expand, but they're being maintained throughout the process of development. And they're there to basically maintain their position. And when the gland becomes mature and it's already fully differentiated, these cells are there and they are forming acini. So these cells in the outer region eventually are already committed to become acinar cells.
Acinar
Cells
cell
in the middle of the
progenitors are
bud
localized
are
in
the
the
cells
outer
that
cell
undergo a lot of
layer.
different rearrangements. They change their identities. They're the ones that become keratin 19 positive. They're the ones that also have a lot of the keratin 5 progenitor expression with SOX2 and some other transcription factors. And these cells, through rearrangements, through regulation by parasympathetic innervation, these cells will become ducts. So the inner parts of the bud become ducts, the outer parts are acinar cells.
Okay, I get it.
So what do we know about salivary gland development?
Did you see they released our
know
spring
that,
schedule in the email?
there
I
are multiple
did. I didn't
regulatory
look at the
factors.
schedule, but it
These
happened
involve
it literally
extrinsic and
happened like
intrinsic
the minute my
factors
stream
that
started.
regulate the development.
Is there anything
And I've
crazy from
listed
it?
some of these for you because
I don't see there's a point of
I
looking
believe
at the
that
schedule right now.
it's better to just have a sort of an outline of all of these factors, and then we can go through and sort of determine, what exactly do they do. There's so much involved in terms of crosstalk. In other words,
You haven't looked yet?
there's
Yeah, I
nothing
saw the
occurs
email.
in a linear fashion, what we know now. Everything is via sort of connected and networks of events. So signaling pathways don't work just one A goes to B, goes to C, goes to D, but in fact, there are a lot of interconnections. And you will see here that some of these components that I've listed actually talk to each other. And when one suppresses an event, the other one can desuppress
Maintenance,
it by
signaling, Sonic
inhibiting that
Hedgehog,
component. So these are these beautiful
EDAR,
orchestrated networks, but it also tells you how
BMP,
complex it is.
TFG,
And
beta.
everything has to be perfectly orchestrated in order to produce this functional and beautifully differentiated gland.
This is relevant
So
to like the
fibroblast growth
last thing we talked
factor
about.
family is most likely the most important.
It is a
family,
great
but at the
day to
same
be
time,
a
epidermal
Mercedes
growth
fan.
factor
Yes,
is
hell
extremely important.
fucking yeah, it
actually
was.
is
Dude,
frequently
Hamilton would have won.
correlating with cancers,
saying.
both in salivary glands and other aspects of oral cavity.
Hamilton would have won.
Wind signaling, you probably heard too much about already.
It's reminding you of
never
exam
stops
one.
being important. It
Never
comes back in
stops
different
being
ways, and the more we
important.
study, the more we find out that it's involved in 10 times more functions than we
See, it's in
thought.
everything, bro.
Hedgehog signaling is very important early in development. Then there's also
WNT.
ectrodysplasia, EDAR signaling, BMP, bone morphogenetic protein. You probably heard about that a lot. But all of these factors are critical.
Do we have to
They're
know
expressed both
these
in the epithelium
factors?
and the mesenchyme. They talk to each other for crosstalks. And we will just go very briefly to understand how they function. At the
You got your
same
Russell win.
time,
Honestly,
we
I'm
have
so
this
happy for him.
neurotropic growth family.
I'm happy for Russell to get
growth
his.
factor
He finally got his
components that are
win
absolutely
after getting
critical
his
because they are the
original one
ones that are
stolen.
communicating with other aspects of the interacting systems from the extracellular components, and these involve the parasympathetic innervation. There's also the neurotransmitter signaling that's very critical. And of course, extracellular matrix protein, you probably all heard about it, right? So you have this epithelium that grows into the mesenchyme, but ultimately there's
Xerostomia,
also
hypoplasia,
ECM,
oral
or extracellular matrix that
infections.
contains collagens, contains fibronectin, contains, here we are looking at laminins mostly because laminin is so critical for salivary gland development. And these components, again, are impacted by what happens in the epithelium. They themselves talk back to the epithelium, but at the same time, if you start perturbing their abundance, perturbing their expression, you end up having messed up development.
And you see how Max doesn't mature.
So,
He's just good at hiding that he's
one
just
of the things
hiding
about our studies with salivary
fact that he's
gland
not mature.
is that we now know that
I've been so
very
mad about
frequently
it.
when we have patients with craniofacial abnormalities,
To be
or
honest.
patients that have actually been diagnosed with maybe some aspects of salivary gland disorders, but these patients also have mutations in identical components to mouse models, okay? So what we can basically derive from some of the studies with mice is that we can make some connections as to anticipating what happens in humans. Unfortunately, salivary glands have not been considered to be very important because people didn't think that saliva was important for a long time. So most of the studies in terms of development of the craniofacial complex comes really to look at the craniofacial abnormalities. However, most of
my God, my
craniofacial
neck.
abnormalities are also associated with salivary gland abnormalities. So I want you to keep this in mind because we definitely have some of the mutations that are recognized for craniofacial mutations, but also have mutations in salivary glands.
I think the bottom part is probably the only thing that's important, and also just knowing a little bit of
so let's go to fibroblast growth factor. This has been
growth factor
studied most
families.
effectively by many investigators over the past maybe 3 decades. And it was shown that it really is a key regulator of this branching morphogenesis. And what I include on the bottom here is this FGF fibroblast growth factor receptor 1.
I would love to know what the 5 questions could possibly be about.
In terms of serving as a receptor for FGF, fibroblast growth factor, mostly F10, fibroblast growth factor 10 and 7. And what is known is that if you start inhibiting this receptor, and this is done in ex vivo system, where you basically dissect a mouse gland with the underlying mesenchyme, and you put it in a dish, and you can grow it with particular supplements and under air liquid interface. And what it does, it undergoes normal branching morphogenesis. You can do it for 3-4 days, and you end up with enormously differentiated gland. But if you treat it with inhibitors of FGFR receptor 1, what happens is you basically have blunted development. And what this is actually shown here, that after a certain period of time, 24 to 48 hours, you will see that the glands actually stop to develop. And this shows you that by this inference, it shows you that this particular receptor activity is absolutely critical for the development of a gland. Now, humans have these mutations also in FGF8 and FGFR receptor 2, which is a receptor for the particular FGF8.
Is important.
And they have multiple craniofacial abnormalities, and we know that cleft lips and palate actually are mutated in these particular components.
FR2.
So the fact that ex vivo we see that there are mutations occurring, not mutations, but defects that we can trace in particular developmental program, that will indicate that in fact this is very critical also for salivary gland development. So we also have an epidermal growth factor family of receptors, and these are EGFR. You probably heard about it. It's one of the key regulator of homeostasis, but it's also a key regulator of tumorigenesis, and it's frequently dysregulated in cancer. And they do, the EGF family of receptors, EGFR, regulate multiple functions that are involved in proliferation and migration, differentiation, and it's all listed here, even the cell death, apoptosis. We know that the salivary glands actually express a number of these different receptors and number of ligands. And the idea is that some of them can have compensatory functions, meaning that if you delete one, you will have able of another factor to compensate for the absence of another. But at the same time, we know that some of them, under some stages of development, they cannot be compensatory. And this is basically dissected out by looking at different stages of development and then inhibiting different components of the EGF receptor family. And then you find that, in fact, under some conditions, you will not have a rescue with other components of EGFR family. And what we do know is that there are major reduced branching defects in salivary glands that are inhibited for this particular growth family receptors.
Anyone start cold?
And ligands during salivary gland development.
The cold of Kyo.
One of the things that is very important here is also that you will see later that some of the parasympathetic innervation is absolutely critical to maintain stem-like activities or progenitor cell activities during development.
Wait, where the hell are we? Are we in
And if you
WNT?
don't have that innervation, you lose some of the progenitor cells. And if you lose progenitor cells, you are also losing the ability of the gland to undergo further development, undergo further production of new cells and lineage basically lineage, following lineage activity, so that these cells will undergo the pathway of differentiation. If you start diminishing these cells, you are not going to generate all of the differentiated cell types.
HBEGF
What we know
activation
that EGF
controls
receptor signaling is very important to mediate this parasympathetic
differentiation.
innervation with maintenance of keratin 5 positive cells and vis-a-vis progenitor cells that mark some of the SOX2 activities as we showed earlier.
Required.
Now, of course, then we go back to Wnt signaling, and I just want to very briefly say, of course, this is a pathway that drives patterning in many organs. It's very critical for salivary gland development. But what's interesting about Wnt signaling is that it can be distributed in different parts of the salivary gland during development. So it is in ducts. It can also be in end buds. But while it drives duct development, it actually inhibits end bud formation. So this is a scenario which is sort of interesting because when you suppress end bud formation, then obviously you cannot expand branching, you cannot cleft anymore, you cannot do any of these processes that are important for functional differentiation of a gland. So what happens there, you have FGF. Remember, FGFs are important for end bud formation. So FGFs will suppress Wnt signaling in those particular areas so that Wnt is not going to be inhibiting end bud formation. So this sort of tells you that there is this crosstalk among different pathways, and these pathways are absolutely tightly regulated, and they talk to each other. And if something gets dysregulated in this crosstalk of activities, then you will not have proper differentiation program.
Mutations in Wnt genes have been associated with...
So we also have ectodysplasia, EDR signaling. This is essential also for salivary gland.
What are dental x-rays called? What are they called?
Morphogenesis, and we do know that mouse models with these particular mutations will mimic a human disease. And this pathway is very important because it's also interactive with Wnt and FGF signaling. And again, there are a lot of different activities that basically depend on each other. So many of these pathways depend on each other, depend on the cross-regulation with each other.
You should try that with the group
and
chat.
Lastly, the BMP family, bone morphogenetic protein. This comes from TGF-beta family.
See how that one will work with the group chat. I want to see their reaction.
Clinic, in the context of dental clinic, because it actually is used to be able to guide some of the wound healing in the oral cavity when surgeries are performed. And so this is very important to be able to understand what are its key functions. And again, salivary gland express many of bone morphogenetic proteins. They are basically driving, again, like Wnt, patterning and affect cell migration and cell differentiation. And so these
that played through my what's it called, my speakers. It scared the hell out of me.
Branching morphogenesis.
Why did that play through my speakers?
So now we are just going to go very briefly. Hedgehog is also important. I just listed it here because it's regulated by FGF pathways. And then finally, this neurotropic growth factor family is critical for innervation, as I said. And this is on the bottom, you see, this is the dissected out gland.
Don't even get me started. Don't even get me started on the whole Checo Perez, Max Verstappen thing. It is wild. It's just crazy that like everyone is just seeing now how he acts.
Like 14, 15 here, 14 maybe. And as you see, it actually is already enveloped. In red, you see already enveloped this parasympathetic innervation. When you remove this innervation, which you can do by basically pulling it off ex vivo in culture, you end up getting loss of keratin 5 positive cells. And this means that innervation is absolutely critical for the maintenance of these immature cells. And if you remove this innervation, the gland cannot expand and it will not develop properly. So I just want to very briefly go through this parasympathetic innervation of a ductal system. And this is actually a study that was carried out in the fairly recent, last decade, that shows how salivary glands actually develop the ducts. So what we do know now is that epithelial buds, as they are being formed, these buds actually secrete a protein called neutrin. And neutrin brings in parasympathetic innervation onto the epithelia to start making sure that these start enveloping the ductal region as the ducts expand. And that's absolutely critical, again, for maintaining some of these early progenitor cells. So what you see here in this cartoon is this branching gland at embryonic day 13. And most of the time you see there are just these green cells, these are keratin 19 immature cells.
Okay.
And for a long time, there was a big controversy as to how do these cells that are forming initial by condensing, forming ducts, how do they become hollow? How do you luminize these cells? So eventually, by tracing some of these cells and looking at the localization and the distribution vis-a-vis each other, we were able to see that they actually form spaces in between, and they form something we call microlumens. And these microlumens are these little holes that are formed in that region, and eventually they will fuse to form a bigger lumen. And this is the process that actually drives this luminization. This was it happens in some other organs, but was not expected to follow this particular pattern in salivary gland. So that was a very interesting part of lumen formation. But what we do know is that you need this parasympathetic innervation in order for this process to start and to proceed. So again, you have these parasympathetic ganglia that are condensing around the ductal cells, and these parasympathetic ganglia will promote these microlumen formation, then condensation of these lumens, and then expansion of lumens to allow the flow, ultimately the flow of fluid as the gland develops.
Sometimes you miss the memories and not the person.
So
that
what else
so sad.
is very important is
that feels like
now, so here we talked
like a breakup
about
kind of
having
quote.
a component
Someone going through
of
it?
the epithelium
Someone
that
going
is
through
going
it
to
here?
be impacting
I hope you're
parasympathetic
okay.
innervation. But we also know that nerves themselves secrete components that are also impacting what's happening in the epithelium. And this is, again, another example of this crosstalk. So we have this nerve-derived factor, which is called vasoactive intestinal peptide. And this actually establishes a communication with the epithelium, and it's also very important to collaborate with neutrin to be able to drive this
I love
process.
That. I like the tooth with the heart.
So again, this duct elongation.
Should I be highlighting? Do you think I should be highlighting and like noting all of the stuff that's in orange? Is that like her way of saying what's important? Okay, so the neurotransmitter signaling is. Oh. Well, it's a good thing I figured that out only fucking 16 out of 22 slides in. Play, pause.
Through proliferation of cells, because the cells grow and they elongate the ducts, then you have fusion of microlumens, and then ultimately luminization to allow flow through. All of this depends on parasympathetic
K19 cells proliferate, forms microlumens. Yep.
Lastly, the last step, I just wanted to very briefly just go through the clefting process. So, as I mentioned, these initial buds that are just basically one ball-like structure, they need to be clefted in order to expand. And so this, as you see, occurs through the particular deposition of proteins that are both on the outside of the bud in the mesenchyme and in the epithelium.
So fibronectin is doing the clefting.
In the mesenchyme, you actually have fibronectin, which is deposited all over, but it's particularly condensed in regions of the bud.
Yo, I need brain cells. Give me some.
And
I was asking a friend.
I put here an arrow where the clefting occurs.
a friend got 100% on our last exam, and I'm all like, dude, give me your brain cells. Let me absorb some of your brain cells, man.
And what does it do? In order to be able to make this cleft go inside, to go internally into the bud, you need to stop cells from adhering to each other, right? And in order to stop this adhesion, you need to inhibit components that are driving cells sticking together. This is called epithelial cadherin or E-cadherin. So the fibronectin deposition inhibits E-cadherin-mediated cell-cell adhesion, and that allows the cleft to form. And if you see, like in part B here, we see that these clefts grow,
I don't
and
know
they
how
grow
much
at
level of
different
detail I
rates
need to
because
know
it
for
depends
that. What
when
the
they
heck?
start getting initiated. But it's fascinating to see whether these clefts actually eventually will lead towards the connection to this growing duct. And then this particular bud that you see as being well defined, that bud is going to itself form a branch and undergo clefting. And that goes on and on and on until about embryonic day 18. Another part, so fibronectin is very critical, but we also know that laminin, which is a basement membrane protein, and it's absolutely critical for salivary gland function.
Laminin 1 and
But laminin 1,
5.
alpha 1, and laminin alpha 5 are also very important in terms of generating regulation of clefts. So laminin is also accumulating.
I guess that's really what we got to know is just
if you delete
fibronectin
laminin,
and then laminin A1, laminin A5.
You're going to find that you have defects in cleft formation. But fibronectin is really the key driver of this. So if you don't have fibronectin, you're not going to have cleft formation. So this is sort of an overall summary of the gland development. And I think I listed here 6 different aspects of salivary gland development, and I would like you to understand those different processes.
Oh shit, is that what it was?
So.
Oh god, that's a lot.
So if you look at the top right hand on my part here, you see end bud formation. So you start with a single epithelial bud, and this bud is going to undergo different processes that involve outgrowth, involve clefting, and then ultimately to connect with ducts. There are progenitor cells, which we discussed, that are absolutely critical to maintain the outgrowth of the cells, to maintain lineages of cells, and to be able to make sure that cells are following a lineage pathway that is prescribed for them. So ductal cells will have a different lineage pathway than acinar cells. Then we talk about lumen formation, and luminization is absolutely important in order to be able to form that cavity in which the saliva is going to flow from acinar buds to the excretory duct and to the oral cavity. We have cleft formation, we just discussed, which is absolutely critical, and it's regulated by many extracellular matrix proteins. Innervation is regulating many different aspects of salivary gland morphogenesis. And of course, duct formation, we just talked about, that also is critical in terms of being able to outgrow and follow up the pattern of budding. So all of it has to be coordinated.
Duct formation is just all of the above.
Of these pathways that are involved. Now, I don't want to have you memorize everything because that's not going to help. Yep. The idea is that you should know that these are the stages of development and that this is a process that is cross-regulated by many different key regulators. And what's really important is the ones that I listed at the very beginning as to what are the key regulators, is to have a sense what they do, and to make to have the understanding that if you have mutations in these components, or if you inhibit them, either by pharmacological inhibition or by basically using molecular approaches like inhibition of messenger RNA via siRNA or the hairpin RNA, you will be able to actually mimic some of the phenotypes that we see in mutations in humans and also in mice.
So we need to know the sample.
So I just wanted to finish by talking a little bit about salivary gland dysfunctions that we know are occurring in humans. And the idea is that we also can use mouse models to understand better how these dysfunctions are regulated, what are the key components that are responsible for some of the morbidity that is associated with these mutations, and also to figure out perhaps one of the interventions or some of the interventions and develop ways in which we can manage these. So one of the key dysfunctions that we know about in the oral cavity of salivary gland is Sjogren's syndrome. And Sjogren's syndrome is an autoimmune disease. And for those of you who, I'm not sure how long ago it was, maybe a decade ago, Venus Williams, who was one of the Williams sisters, tennis player, she was actually participating in US Open. And at one point, she had to withdraw because she was diagnosed with Sjogren's syndrome. And although it doesn't seem to be such an amazingly bad disease, in fact, it has huge association with morbidity. And if it progresses, it actually can be a basis for development of B-cell lymphoma. So it is a very severe disease. But the problem is that most people who have this disorder are not even diagnosed with it because there are not known many molecular features of this disease that we can actually follow up and diagnose patients accordingly. So for instance, if you have an individual who comes and has a dysfunction, let's say of salivary flow, and they will complain that they don't produce enough saliva, you will typically chalk it up to either, some sort of, stressful situation in the life or with age. It can happen also, older people sometimes do need to have, some supplements for saliva. But typically, you will not diagnose these individuals with disease. There are particular criteria that are now generated by European and American systems. And these criteria involve looking at a particular foci of immune cells, because as you imagine, the autoimmune disease means that you have like a recognition of some of the aspects of your gland that are considered to be non-self by the immune system, and they will get destroyed. And so ultimately what happens with patients that have this disease, you are actually losing some of the salivary gland and you're losing the functions. In addition, these are very much connected to lacrimal glands, to the secretion of tears. So both dry eyes and dry mouth are a typical example of the Sjogren's syndrome manifestation. So what actually we know now is that there are a lot of defects, and by using different molecular approaches, we are able to try to figure out what's happening with these individuals. So we actually, sort of in my lab, we ended up actually collaborating with a group in Norway, and these individuals have a very high actually recruitment of individuals who have dysfunctions in salivary flow, and they diagnose patients with Sjogren's syndrome much more in Scandinavia than they do in the United States. And so what we did is we ended up actually getting glands from patients, and we examined them for gene expression signatures. What we did is we wanted to know what was the global transcriptome of these patients. And by doing this, this is sort of shown on the figure here, in the figure here, we were able to show that in fact there is also so-called the mixed gene signature phenotype, where patients who have already some indications of gene signatures that are perturbed and found in Sjogren patients are also found in patients who are not considered to be diagnosed with this disease. And so what this might tell you is that perhaps if we can do these kind of studies and further expand them, we can tell who is going to be prone towards moving towards this disease. And the same thing happens actually in cancer. We are now looking, there's a huge effort to look at pre-cancer stages, so that if you see some lesion that's not considered to be malignant, you are just going to be left with it and they will be watching you. But the fact is that some of them will flip and become cancer. So how can we tell whether some of these lesions can actually be driven towards disease states? And by doing some of these molecular analyses, we believe that we can actually decipher some of these situations and maybe be able to stratify patients based on what they express. So this is sort of one example, and also I wanted to just show you that here's this E-cadherin adhesion molecule. This molecule is very important in driving cell polarity, and it's important for maintenance of polarity. It's also a tumor suppressor. But if you look on the bottom part here, this is what it looks like, the tissue looks like from Sjogren patients, and you see that the cells are basically losing the polarity altogether. And so because they stop expressing this particular cell adhesion molecule, so these people have totally messed up structural components of cell identities. So the next thing I want to tell you about is a little bit about salivary gland neoplasms. So these are the cancers of salivary glands, and you may be surprised to know that there are at least like 25 different histopathologically different cancers of salivary glands, and they're very bad for you. I mean, they're just really dangerous diseases. They constitute only 3 to 6% of head and neck cancers, but when one gets this particular disease, it's really difficult to manage clinically. And in fact, there are almost no clinical strategies to manage these particular tumors. So I just listed some of them for you here. So they include like adenocystic carcinoma. In fact, we have one of our colleagues used to be a financier. He was an investment banker. And then his wife was diagnosed with adenocystic carcinoma, and he dropped out of working in the banking system, and he set up a foundation. And his foundation is called Adenocystic Carcinoma Foundation. And his name is Jeffrey Kaufman, and he's actually become extremely vocal and very active in the field to try to understand how these glands become dysregulated. And these are submandibular glands, parotid glands that can become actually turn into tumors. So one of the things I wanted to point out, and you have it also in the early stages, early slides that I included, is this structure of a salivary gland. And if you see, you know, you have these serous acini, which are basically producing a lot of proteins. Then you have, you know, mucous acini that producing mucins. And then there are the serous demulin, which are the aspects of part of the mixed composition of salivary gland, and which is what we find in submandibular glands, but also produce more proteinaceous components. But if you look at the ductal system, you see that they're basically lined, especially intercalated ducts, are lined with single cells. In other words, there's one layer of cells. But as you move further down, you see that, in fact, there are some of these cells that are facing the lumen, and they have obviously, I hope I didn't stress it, but the side of the cell that is differentiated, that faces the lumen, is apical, and the side of the cell that is not facing the lumen and is more towards the matrix is called the basal component.
Yep.
But there are also basal cells there. So now these are these additional cell types there that are absolutely important because they are the ones that can also proliferate and maintain some of the immature status of the glands. So these are the ones that can undergo changes in identities. They can expand when they're not supposed to be expanding, and these are the cells that frequently drive carcinogenic process. Another cell type that I actually didn't mention, but is absolutely critical, is myoepithelial cells. So in order to secrete, in addition to having all of the parasympathetic innervation, you need a communication with the another cell type, this myoepithelial cell. It is actually rich in actin-myosin structures, and these provide contractile function. And in order to secrete, you need to contract, right? You need to contract the gland. So myoepithelial cells are also the cells that can flip very frequently, and they can undergo transformation to carcinogenic state. And the difficulty with this is that these cells don't just become another cell. They can become one cell, another cell, third cell, fourth cell. They change identities very frequently. And this is what happens in neoplasms, that we have new cell identities. And these actually, the changes in cell identities, or we call it cell plasticity, is actually one of the major features or hallmarks of cancer. And most recently, there's a new review on cancer and hallmarks of cancer, and it has been recognized as one of the key factors, new factors that are driving the process of tumorigenesis and carcinogenesis in general, and vis-a-vis cell plasticity. And so what we hope is when we can regulate these cell types, we can stop them from losing identities, from acquiring new identities, and understanding how to target these cells. Perhaps you are going to be able to inhibit some of these tumors that are forming. But it's a big problem, and clearly, you know, there are not too many cases, but the cases are bad. And one of the things I wanted to mention to you, remember that parasympathetic innervation is definitely a component of salivary gland morphology. And so what happens to these tumors? These tumors migrate to the brain. because of this nervous system that is intercollecting and it's interacting with salivary glands, it provides a pathway of which cells can migrate once they detach from primary tumor, and they can migrate to the brain. So very frequently individuals who have these disorders, these are rare cancers, but the salivary gland cancers can be very, very bad in terms of sequela of, I'm done. So anyway, so at this point, I'm done, and I want to thank you. And if you have any questions, please ask. And
She said, yeah, that's it. What the heck? All right, let's pull up the next one. How long is this one? Oh my goodness, this is a 2-hour one? Oh no. Wait. Oh man, this is a long one. I don't know if I want to watch 3 whole lectures. I'm starting to die. Alright, let's see. Let me pull up this PDF. Yukon. Goodness gracious.
Really interesting developmental problems, and so you'll get a chance to see images of some of these developmental mutations and growth defects, and it's really
I can
fascinating
barely
area.
hear.
So please welcome Dr. Reichenberger.
Hi, good afternoon. So thank you for the introduction. And so I'm going to show you, or to guide you through a number of craniofacial malformations that you as dentists, user dentists, will come
Interesting.
across most likely. And so why is it important for you? Because you will need to treat the patients. You will need to know what the cause of the disorder is, and which treatment would be good or would not be recommendable. So the groups of disorders that I'm going to cover are the cleft-things, then craniosynostosis, some developmental abnormalities, and in our lab we work with disorders of increased bone deposition that's limited to the skull bones, and excessive bone resorption that's limited to the skull bones. And the whole research of the molecular origins of diseases that started only like 120 years ago. It started with physicians and biochemists, and now, then over the time, there were bioinformatics people involved when all the genetic mapping was done. So that's largely finished now, and now it's molecular biologists, it's physicians and dentists, and I will show you some examples where dentists become important to do the functional studies of craniofacial disorders. And as you probably heard many times, this is the era of personalized medicine. So we want to know the genetic origin of the disease.
Why did my VTube studio crash?
Patient, to help with the diagnosis and the treatment, and in the future even more with preventing diseases. And there are so many craniofacial malformations. So
this guy sounds like Total Wolf.
More than 10,000 syndromes known to science, and 7,000 of them are kind of rare disorders, depending on how you define rare. Usually it's diseases with less than like 200,000 patients at any given time. And out of these 7,000, there's about 1,700 that are monogenic, and others are maybe sporadic disorders, or they are multifactorial, so meaning they have several disease genes that need to play together. And what's important to know is in the US, out of about 4 million children who are born every year, about 7% have some kind of physical or mental condition. And 5% of these 4 million, which is about 200,000 kids, they have some kind of disorder that involves craniofacial malformations. And worldwide, obviously, it affects millions of people. So all the rare diseases together is not really rare, right? But it's very difficult to treat, to assess, because many physicians, dentists, they never see all of the disorders that are out there, and all of a sudden somebody comes to your clinic and you don't know what to do with them. So another principle I want to point out here is that many genes that are active during craniofacial formation also are active in other organs and in limb formation. So oftentimes you see craniofacial malformations syndromic with other symptoms. So why are craniofacial malformations so common? One reason is that the development from a very early developmental stage to a finished facial features takes about 4 or 5 weeks. So this is in humans. In mice, it's only a few days. So it's crucially important that all the procedures and the gene expression that is needed is very well organized. So if, for example, some gene is overexpressed, underexpressed, expressed too late, then that already can, during this 4-week period of time, then that can already lead to some major craniofacial defect. And so what we have here is, like, for example, structures like the first branchial arch that forms the mandible. I'm sure you're all familiar with that. These structures need to first develop, then fuse. Then the facial structures, they all need to, they all have their origin in the neural crest cells, that have to migrate from the hindbrain to the facial areas, and then there's a lot of fusion of features here, the nasal placoid. All these structures need to fuse. If any of these procedures goes wrong, maybe because of environmental effects, some poisons, the mom has to take some drugs during this time point, that all can lead to craniofacial malformations. So just to go back one more step, I'm sure you're all familiar with that. Let me just mention it briefly. The monogenic or Mendelian disorders, they have one disease gene that leads to a specific disease phenotype. Some family members may be more or less affected, and that's due to modifier genes that interact with this disease gene. And on the other hand, you have the complex disorders, where you need several disease susceptibility genes. So maybe, let's say there are 10 genes known, and you need to have at least 5 or 6 in order to express this disorder. And environment and lifestyle, epigenetics are also very important to develop this complex phenotype. And one of these complex disorders is clefting. So you can already tell that it's most likely a complex disorder because it's less common in Africans, in individuals of African descent, more common in whites and Asians. Oh. And so clefting of the lip or the palate is actually associated with more than 200 disorders. It's syndromic with those disorders. And I just would like to show you some examples. So this is an isolated cleft lip. Now in this baby you see a unilateral cleft palate. And unilateral cleft palates are about twice as common as bilateral clefting. And interestingly, in boys, the unilateral cleft palate is about 1.7 times more common than in girls. So there's a lot of factors that are playing into this cleft formation. And the way it looks like in the mouse, in this schematic you can see a cross-section through a 11-day-old mouse.
Bro, that's not a mouse. That's like a weasel.
In the oral cavity, the palatal shelves are still undeveloped or underdeveloped. And what happens now is that the palatal shelves, they need to grow down and then elevate and then fuse. So for this part of the development, there are some crucial genes that you should remember because they come up in many other disorders as well. For example, the bone morphogenetic proteins, in this case BMP2, then FGF receptors, fibroblast growth factor receptors and fibroblast growth factors. Then there are a number of homeobox genes, for example MSX1, that's crucial. And all of these direct the palatal shelf growth and the elevation. If the mom takes, for example, statin drugs, that could interfere with the expression of these genes and lead to clefting. Or the mom takes, for example, retinoic acid cream against acne, not knowing that she's pregnant. And that was a big problem in the 80s when this effect was still unknown. That many kids, when the mom took Accutane, they ended up with clefting, but only during this 4-week time period. And then after the shelves moved up, they need to fuse via epithelial mesenchymal transition. I'll talk to you about it more in the next slide. And if that doesn't happen because of some drug or environmental interaction, then the baby may or may not have the cleft. And the only gene that they need to differentiate into mesenchymal-like cells. That's called epithelial mesenchymal transition. So they lose gene expression that's typical for epithelial cells and gain some gene expression profile that's typical for mesenchymal cells. And if this step doesn't happen, and this is largely driven by TGF-beta, then there's incomplete fusion of the palatal shelf. So this IRF6 causes, if there are mutations that cause this Van der Bode syndrome, which is characterized by clefting and by these very specific dimples in the lower lip. And there's more than 200 mutations known in this IRF6 gene that lead to haploinsufficiency. That means the mutant protein is not functional or gets degraded. So on a molecular level, I just very superficially want to tell you that this, the loss of IRF6 alters BMP2, BMP4, sonic hedgehog, and amongst others, FGF10. Now this mutation in this IRF6 can also be syndromic with one other disorder, that's popliteal pterygium syndrome, and that's the same phenotype here, together with syndactyly and a webbed knee. So during embryonic development, all the arms and the knee and the legs, they are all joined with a web, and BMPs are some of these molecules that organize the cell death so that this webbing disappears, and so it doesn't disappear in this syndrome. Another important thing that I mentioned
I feel
is
like I'm
MSX1.
about to pass out. Oh my god.
And this is just a risk factor for clefting. So MSX1 mutations by themselves don't necessarily cause clefting, but a specific mutation that causes this so-called Witkop syndrome or tooth-nail syndrome. It also has, specific for this syndrome is like the peg-shaped teeth and this underdeveloped toenails. And many of these kids also have clefts. And that's because this MSX1, as I mentioned before, affects the EMT, the epithelial mesenchymal interaction, and that affects then again DMP2 for sonic hedgehog. There are many other risk factors. Many of them are not known yet. So there are many gene loci that were found by GWAS, but it's unknown what the mutation in this gene loci is yet. Besides that, there are rare facial clefts, and although many of them are sporadic, some of them are hereditary, like for example in Treacher-Collins syndrome or Goldenhar syndrome. And Goldenhar syndrome is part of the spectrum of hemifacial microsomia. So that's uneven development of facial features. So the left side or the right side may be longer or shorter than the other side. And this also comes sometimes with lateral facial clefts, like in this child. And you can also see a little bit in these kids. And the other thing that's characteristic for this unilateral facial hypoplasia is that there are malformed or malpositioned ears. So another disorder I would like to introduce is a monogenic disorder, Treacher-Collins syndrome. I don't know if any of you have heard of Treacher-Collins. No, it's not really so rare. I think it's 1 in 50,000 kids are born with Treacher-Collins. And it's characterized by downward slanting of the eyes and coloboma of the lower lid. Coloboma means it's incomplete fusion of the lower lid. These kids also have very small lower jaws, and another characteristic is the hypoplastic psychomatic arches. The mutation is usually in one gene, that's TCOF1, which is a nucleolar trafficking gene. And in this family, actually, I want you to guess, Treacher-Collins is inherited, and who, the dad or the mom, who do you think has the disease? The mom, yep, right.
Huh?
And as you can see,
How?
the expressivity in the kids is very, very different.
How do you tell?
And that's due to modifier genes, typically. So what happens in this Treacher-Collins syndrome is that this TCOF1 is important for neural crest cell development and migration. And so here we see a heterozygous mouse model. This mouse has a knock-in mutation for one of these TCOF1 for these Treasure-Collins mutations. And what you see in green are neural crest cells. They're labeled with green fluorescent protein. So they light up under fluorescent light. So these neural crest cells, they come from the hindbrain, migrate to the forebrain and to the frontonasal black coat, the area that forms the face. And if there are not enough cells or if they are too slow migrating, then you have part of your facial bones either missing or malformed. And this protein regulates the ribosome biogenesis, so energy mechanisms. And it also functions in DNA damage response, and it works, it counters p53 activation. So in the mouse model, and so p53 is a trigger for cell death. And in this mouse model, what they did is they inhibited p53 and could rescue the phenotype in the mouse. Obviously, you can't do that in humans because so many other things would get messed up. And in this gene, there are also like more than 200 mutations that lead to this, that lead actually to premature stop codons and loss of function. And in the mouse, it's estimated that there are like only 3/4 of the neural crest cells are able to migrate. And the consequences that has in humans, it can be devastating, like you see in this girl. This little girl has Treasure-Collins syndrome, and you can actually Google her, don't do it now, but you can Google her under baby Juliana. She lives in Florida, and she misses like 30 to 40% of her facial bones.
Wow.
So now she's about 16 years old, and she seems to do very well, and that's her sister. And the family actually adopted another child with Treasure-Collins a few years ago. So needless to say that she had many, many surgeries to get to the stage that she's now. So, the next disorder I would like to touch on is craniosynostosis, which is a monogenic disorder. And that's the premature closure of calvarial sutures.
What the heck? That's
here
crazy.
You see all the sutures open, and here you see one of the coronal sutures closed. And this is also fairly common. There's about 1 in 3,000 kids have craniosynostosis in one form or the other, and it's syndromic with more than 100 disorders. So a very mild form would be this, what's called trigonocephaly. So the metopic suture fuses prematurely, and so this kid, you can see it, but this kid is probably doing pretty well for the rest of his life. And what's happening on a molecular basis, so here you can see all the sutures. I'm sure you're familiar with that from your anatomy class. The metopic, the coronal, the sagittal, lambda sutures. And what's happening here is that mutations in or abnormal expression in some of the genes that I mentioned before, like homeobox gene, BMPs, FGFs, can mess up the normal positional growth of the cranial plate. So here you see cranial bone. And cranial bone, as you know, does not grow by endochondral growth like the long bones, but by appositional growth, right? So the new osteoid is, and the bone cells, they divide at the tip of the cranial plate, and they form this unmineralized bone, which later mineralizes. And the job of the cranial suture mesenchyme is to keep these bony plates apart. So as soon as and that's you see here in orange, as soon as this suture mesenchyme is affected and disappears, the cranial bones, they fuse, and you end up with craniosynostosis. So this suture mesenchyme consists mostly of stem cells, and genes like FGF, MSX2, and some others are crucial for keeping these stem cells in a stem cell stage. As soon as they differentiate, they may become like osteoblasts, and you have complete fusion of your suture. Okay. So, and by the way, if you have any questions, you can interrupt me at any time.
I should just rewind that, to be honest.
You see a child with a very severe form of craniosynostosis. This is called cloverleaf skull, and this is caused by a single mutation in this MSX2 gene. So this mutation leads to a single amino acid change, and that causes the the protein, the mutant protein, to be degraded very, very quickly. And the degradation goes, this is one of the canonical degradation pathways, by proteasomal degradation. I don't want to go into the biology of that. So what the only treatment for these kids would be to take all the single cranial plates out and put them back together with like a kind of a spring, so the sutures can stay open and can keep expanding. Otherwise, there would be increased pressure in the brain, and the child would not survive for very long. And as I mentioned before, this MSX2, it's important for the suture maintenance, and if there's not enough of this MSX2, you get degradation of this stem cell mesenchyme. Another important gene or group of genes for craniosynostosis are the fibroblast growth factor receptors. So here you see FGF receptor 1, 2, and 3, the extracellular IgG-like domain, the transmembrane domain, and the intracellular active tyrosine kinase domain. So depending where the mutation is, for example, the yellow, the Apert syndrome, the blue cluster, Cruzon syndrome, depending where the mutation is, you get very, very different disorders. And the green one, thanatophoric dysplasia, it's actually lethal. So here is a child with Cruzon syndrome. That's autosomal dominant, as I mentioned before, for all craniosynostosis types. And what's characteristic is the hypoplastic maxilla, so the recessed midface area, and the bulging eye, because the eye sockets are not don't fully expand and develop. And there's one group of mutations responsible for that. And if the mutation is, for example, in the FGF receptor 3, then these kids, it's a specific mutation, then these kids also get Cruzon syndrome, but with acanthosis, what's called acanthosis nigricans, and that's like darkening of the skin in the axilla and around the eyes. And that's because FGF3 is an important factor in skin as well. And so it's, if the mutation is there, it's syndromic with some skin phenotype. So the recessed midface, actually, it's due to a disturbance in the synchondrosis of the cranial base. You see that here, the spheno-occipital synchondrosis. Do you remember that from anatomy?
Nope.
Or you didn't?
Nope.
You didn't have that yet. Oh, okay. Well, then remember it for anatomy, then you can look at it more closely. This synchondrosis, it's cartilage, and it has to stay open for like 7 years so that the midface can grow out together with the growth of the skull. If there's a mutation in the the sutures, then it also has effect on this spheno-occipital synchondrosis. So it mineralizes early and doesn't expand. So Apert syndrome, I mentioned that. The phenotype, the facial phenotype is similar to Cruzon syndrome, except that it's more severe, can be much more severe. So here you see, in this kid, you see the bulging eye because the eye socket is really, really shallow, and you see some extreme midface hypoplasia. And on this side, you can see the craniosynostosis, the fused suture. And I mean, this gives you an impression how much of the, and that's actually a younger kid, that's probably like a maybe 2-year-old kid. And you can see how important it is to keep large parts of the cranium unmineralized for proper growth. However, Apert syndrome, with Apert syndrome, some of the kids also have clefting of the secondary palate. And with this Apert syndrome also comes some disturbance of the fingers and toes, so you get syndactyly of fingers and toes, and so that leads to what's called a single toenail in some more severe cases. So that's because this FGF2 has, or this specific region of FGF2 is also very important in organizing cell death between fingers and toes. Now let me come to some disorders that we investigate in our lab. And one of this is called craniometaphyseal dysplasia, or CMD. And that's a disorder that disturbs the bone homeostasis in skull bones. So here we have hyperostosis, increased deposition of bone. And on the other side, we investigate a disorder that's called cherubism. And in cherubism, you have very severe and very aggressive degradation of bone, resorption of bone by osteoclasts. So they're two very opposing phenotypes. So CMD is a group of what's called craniotubular bone dysplasias. So cranio, because of the skull, tubular, because it also affects long bones to some extent, but the crucial part is the cranial part. And it's a whole group of disorders that fall within this category, but we just focus on CMD here. And there's two forms. There's about 20 years ago, we found the disease mutation for the dominant form, and a few years ago, we found the disease mutation for the recessive form in aconnexin 43. So the dominant form, it's in a gene called ANC, and I'm going to show you that in a minute. So the facial phenotype of these kids is very characteristic. So you have these kids have a wide nasal bridge, and they have craniofacial hyperostosis, mainly in the jaws and also in the cranial base. So sometimes this cranial bone can be like up to 2 centimeters thick. And on the other hand, you have some effect on long bones, especially on the metaphysis of the long bone. And these metaphyses are undertrabeculated, and probably for mechanical reasons, like in osteogenesis imperfecta would be very similar, because there's not enough stability. The bones kind of widen to make up for a more stable bone. And there's very little trabecular bone in this metaphysis. So the mutation I mentioned is in this gene called ANC. ANC is a transmembrane protein.
I'm gonna take a break after this
well, for the
this
longest
first
time, we
hour.
believed it transports pyrophosphate. And
I can't
pyrophosphate
focus
is
anymore. I'm
very
so
important
tired.
to regulate mineralization of bone. So the balance between phosphate and pyrophosphate needs to be very tightly regulated. Otherwise, if you have if you don't have enough pyrophosphate, you get very, very hard bone. If you have too much pyrophosphate, you probably get very soft bone. And pyrophosphate is important actually in all cells of the body, and in toothpaste, I should say. And what it does in other cells, it prevents mineralization. So that your liver does mineralize, the brain, very important, it's highly expressed in brain, does mineralize. And it's an important part of toothpaste because it prevents plaque formation, right? But what we recently learned from some publication a year and two ago is that it also, and maybe even more so, transports ATP and citrate. So ATP and citrate are molecules that are important for the energy cells, for the cellular energy metabolism, right? If you remember the Krebs cycle, no? I didn't look it up.
What's Krebs cycle, JK?
What we found is that all of these mutations here are within a certain region of the molecule, whereas other mutations, they don't have any effect. There's another disorder, chondrocalcinosis, that has mutations here, but I'm not going into that today. And the effect, and obviously you can't do any of these studies in humans. You can take X-rays, you can do CTs, you can do blood work from humans, but it's almost impossible to get like bone tissue unless they have surgery, which they actually do need because the mandibles, they get so massive that they have problems chewing. The nasal passage gets closed in with bone, and the kids can't breathe, they can't drink. That's when when the babies can't suckle, then that's when it usually gets diagnosed. And also, what's even worse is that the cranial foramina get closed in, and that squishes the nerves. These kids, they get deaf, they get blind, and they get facial palsy, and so that's a big problem for them. Probably the worst problem for them. And so what we can do is we can transfer the mutation into mice, into so-called knock-in mice. And here we investigate homozygous knock-in mice, although this is a dominant disorder. But in heterozygous mice, we see a similar phenotype, but it takes about a year to develop. In homozygous mice, this phenotype is visible immediately. And in the wild-type mouse, what we see is very nicely structured bone. This is actually the apical end of the incisor in the mouse. That's the incisor is the tooth that keeps growing lifelong in all rodents. And if you take a higher magnification, you see this very nicely structured bone. In the knock-in mice, on the other hand, you see this really dense bone with all these holes in them. And what we think is these holes comes from osteoclasts that sit there and may resorb bone, but the osteoblasts are unable to remodel. And in higher magnification, you see how ill-structured the bone looks like in comparison to the wild type. And in the mice, we also see a very massive jaw bones, as if you compare the wild type to the knock-in mouse here. So they do replicate the human phenotype. And the other thing we see is that in wild-type mice, we have nice, big osteoclasts that we can grow in culture.
Oh, like the radial
opacity. In the knock-in
mouse,
we see very few osteoclasts. They are, the same number of precursors,
it's
but
really
these precursors,
thick.
they don't want to fuse. And if they fuse, they don't want to move around very much. So that's one of the major phenotypes that we developed, that we found early. And here, again, I want to show you osteoclasts in red after a staining that's called trap staining. Nice osteoclasts with a cervical loop and the stem cells that are forming the incisor. And in the knock-in mouse, you see very few osteoclasts and the messed up structure. Then when we did some molecular studies, we looked at osteoclasts from the calvarium, and we looked at osteoblasts from the femur, and we saw that the knock-in mice don't seem to have any protein. So what happens is that they do make the protein, but the protein gets degraded very, very quickly by this proteasomal degradation mechanism. And so here I just want to show you very briefly, these are some osteoblasts. They express the UNC protein in green. These are wild type. The mutant osteoblasts, they don't express, but they don't show any protein. Protein gets degraded very quickly. So when we inhibit the proteasomal degradation with a factor that's called MG132, but that's not important what it's called, we see in the wild type that, yes, we have nice UNC protein in the osteoblasts, and in the mutant we have, in this case, even more. So that was the proof for us that the UNC gets degraded prematurely, very quickly, and probably some of the healthy UNC gets also degraded at the same time. So we did this experiment because we thought, well, if we can stop proteasomal degradation, maybe that would be a way to help people maintain a higher level of UNC protein, and they would not get such a severe disease phenotype. However, and MG132 is actually used, there's a clinical use for it, so it would be possible, but it would be impractical to do that in children, in growing children, and you would need to do that for the rest of their life, and so there would be too many side effects. So this is not a treatment that we can pursue. But recently, we did some proteomic studies, and we found that this UNC protein, if it's mutant, it's only one mutation, and actually that leads, in this case, the mutation leads to the removal of one amino acid. And this mutant UNC protein gains some new functions, and that was very unexpected. So the new function is that this UNC, and not the healthy UNC, can bind to a proton pump, and it can bind to a metabolic regulator, actually the master regulator for metabolism, that's AMPK. And,
it speeds up the metabolism.
That was totally unexpected because we thought, okay, it's a bone disease and something is wrong with the osteoclasts, but we didn't realize that it's really a metabolic disorder. And it's a metabolic disorder that has abnormal distribution of ATP, as probably other things too. And that leads to the dysfunction of the osteoclasts. Probably other things also, but other disturbances maybe can be compensated, but in osteoclasts it cannot be compensated.
That makes sense.
So what we also saw that the mice, the homozygous mice, they're actually smaller, and they otherwise they look okay. They don't live all that long, but otherwise they're okay. But when we did some metabolism studies, we found that they move less and eat more, but yes, they have less fat and they are smaller, and so they have increased energy expenditure because of this dysregulation of this AMPK. So when the one thing to do would be to regulate this AMPK, and that's something that for other metabolic disorders has been done before. And so what we did is we did osteoclast culture, and this one here is untreated osteoclasts. So you see very small, what we think is osteoclasts or precursors, and then we treat with this specific factor, and we see that when AMPK is inhibited, that these osteoclasts form very nicely, very nice big osteoclasts. So what we investigate now is how can we help these kids? Can we regulate the metabolism in one way? So for one way could be that we lower the amount of phosphate that they eat. So with a low phosphate diet, we definitely tell them don't drink Coke and don't drink beer, and don't buy too much McDonald's because this is all processed food that's high in phosphates for several reasons, mostly as preservative. Or another way would be to look at the phosphate-pyrophosphate ratio and whether we can change that. So there are some enzymes other than ANC that interact with pyrophosphate, that synthesize pyrophosphates outside of the cell actually. And can we do any change there? So this will be an enzyme replacement therapy. And that we have mice growing now where we try whether we can do some enzyme replacement or add more of these other enzymes so that we can change the ratio so that the bone and the osteoclast would behave normally.
Okay.
So that's for excessive bone deposition. Excessive bone resorption would be the cherubism disorder that I mentioned before.
Excessive bone resorption, cherubism.
And very typical for the features of these kids is this really looks like a swollen, puffy face, and that's due to excessive bone resorption only in the jaws, maxilla and mandible. And what you see here in this CT is these cavities that are caused by excessive bone resorption by osteoclasts fills in with some fibrous tissue, and this fibrous tissue keeps growing like a tumor and pushes the bones kind of apart. And here you just see the cortical shell is still left of this jaw. Actually, this tumor, it's benign tumor, can also grow into the orbital floor and lead to the upward tilted eyes. That's where the name cherubism comes from, like from the cherubs of the Renaissance. In the Renaissance churches, you see these cherubs, they all look heavenward. And actually, most often, this disorder is discovered by dentists, because the kids like are 2 years, and I didn't say that actually. This cherubism develops first when the kids are like between 2 and 6 years old, and that's the time when they go to the dentist first time. You see this some tooth displacement, and you take an X-ray, and you see all these bilateral cavities in the mandible and maxilla. And then, if you didn't attend this lecture, you don't know what it is, right? But now you know. And on a histology
still don't know
level,
what's going on.
Normal bone. And osteoclasts, they are where they're supposed to be at the bone. They resorb the bone, and the osteoclasts are supposed to come in and remodel the bone and deposit more bone and to keep the bone turnover going. But what happens here is the osteoclasts are so aggressive, and for some reason the osteoblasts, probably the osteoblasts, they differentiate into this fibrous tumor tissue. And here we can see some osteoclasts that have nothing to do here. Normally you wouldn't find them in, outside the bone, and they also differentiate in this fibrous tissue, indicating to us that there's probably a lot of inflammation going on, because inflammatory factors like TNF-alpha, for example, or SIC or others, they stimulate osteoclast formation under inflammatory conditions. And also about 20 years ago, we found the mutation in this SH3BP2. And as I mentioned, this is adaptive protein, and adaptive proteins, their function is to just be there and attach other proteins, bring them from the cell membrane to the nucleus or vice versa. And in different cell types, this SH3BP2 has different functions, but it's an important regulator in most immune cells that we know. And all these mutations actually are within 6 amino acids, and that always puzzled us. Why is that?
So it's
what
caused
you see
by
in
that.
Bone resorption here is the trabecular bone of the femur, in wild type, heterozygous, and homozygous mice. And
What am I supposed to take away from this? Like, give me the main stuff.
Of the femur of the homozygous mouse, you see how eaten up that already is almost non-existing, this bone. And when you look at the osteoclasts in wild type, you see very nice osteoclasts here and there. In heterozygous cherubism mice, you see more of these osteoclasts and really big, aggressively resorbing osteoclasts in these homozygous mice. So why is that? So this SH3BP2, as I mentioned, transports
it gets bigger as you get more
molecules like in osteoclasts, like SIC, for example. You don't really need to remember that name. If there's too much of this SH3BP2, then that's what we see in this western blot. Here you see wild type, and here you see the homozygous. There's tremendous amount of SH3BP2 compared to the wild type. And so why is that? So obviously there is no degradation. And the mechanism for this degradation was unknown for the longest time. So what needs to happen is this SH3BP2 here needs to be recognized.
I don't think we have to know the degradation, do we?
And then it gets slated for degradation. Ubiquitins get attached to it, and then this complex enters the proteasomal degradation that I already mentioned on several occasions today. And what happens in the mutant SH3BP2 is that this ankyrin cannot cannot recognize because of the mutation in this 6 amino acid sequence. So this SF3BP2 accumulates and cannot be degraded, and that leads to this increased osteoclast formation and also increased TNF alpha production. So in the mouse, we don't just see this high bone resorption in the facial bones, but in the entire skeleton. And what I just briefly want to show here, because we already did several rescue attempts, for example, by blocking TNF alpha, so to lower inflammation. So if you block TNF alpha with low dose of blocker, you don't see any difference. These are the cavarial sutures of the mouse. But if you have with high dose treatment, you see real improvement. But if once you stop, then the resorption starts again. So,
Internacept.
there's another attempt was to block with an inhibitor of this SYK protein, which is a tyrosine kinase. And you don't need to look at all of these. I just want to show this is a healthy leg joint. This is the tibia. And in the mutant, in the homozygous, you see this tremendous degradation, and it says DMSO because the blocker is dissolved in DMSO. This is just the carrier, this is the control. And if we block this SYK, this inducer of osteoclast formation, you can completely stop cherubism in the mouse. But this is nothing, again, that you want to do. This is proof of principle, but it's nothing you want to do in little children. And so therefore, we were looking at another phenomenon, and that is that in the mouse, we don't really see this tumor formation in the jaws. So we see some fibrosis all over the body, some inflammation, but not specific in the jaws. And we did want a model where we can study, where we can induce and study the cherubism formation in the jaws. So that's where another dentist comes in, Dr. Yibing Chen. We share the lab. And she's an endodontist. And for some time we thought, well, what if we challenge the mandible somehow? And so there were different ways you can do that. And then we said, okay, why don't we drill a hole? Why don't we do endodontic treatment on the mouse and let bacteria infiltrate and see what happens? Because that's maybe micro damage could be one reason that cherubism occurs in children, because
jawbone
children
expansion.
have the deciduous teeth, and they migrate when the kids are like between 2 and 6 years old, and so you have a lot of bone resorption, a lot of like inflammatory signals because of that, and so this may be a way to create cherubism in the mouse as well. So what she did is, in a wild-type mouse, you do endodontic drilling, and you see and there's pulp exposure, and then you see infiltration of neutrophils, and so this is like you get kind of apical exposure, the apical bone loss, and if you do that in the knock-in mouse, after 3 days, you see quite a bit more of
a lot of
neutrophils
inflammation,
and after 2 weeks, there's huge amounts of neutrophils, and that's the beginning of of inflammation actually. And what we see when we take X-rays of the mandible, this is a section through the mandible, this is the incisor, the enamel of the incisor. What we see is that in the homozygous mouse, this tremendous expansion of the incisor. So now we have a mouse model, because of a dentist, we have a mouse model that we can control the location of the onset of cherubism, and we can control and we can study like every day what happens in this mouse. What's
going
the
on?
initiators of cherubism in the mouse model, and that's something that we're investigating right now. And
still
the
investigating.
question is, why does cherubism only occur in humans and not in other animals? We don't know. Probably because humans have deciduous teeth, and what triggers the cherubism? It could be the tissue damage, or it could be microorganisms. Because microorganisms and tissue damage, it's called pathogen associated molecular patterns and damage associated molecular patterns. So both of these, they act through a toll-like receptor.
I feel like this is the only slide I need to know.
In the case of macrophages or osteoclasts, they act also involve SYK phosphorylation and SF3BP2, and that could lead to increased TNF alpha production and other cytokines that are pro-inflammatory and leads to the jaw-specific inflammation. So maybe dentists may recommend better oral hygiene to lower the inflammation, or they prescribe antibiotics, and I see I need to finish here, to keep cherubism down. So that's something we're trying to work on for treatment. And of course, there are many other rare disorders that we could talk about, but I think I just covered some of the major groups here. Thank you.
Hooray! Alright, that's 2 down, but I am so sleepy. I'm just gonna go ahead and just turn this into a part 2. I am like really tired. I think I just don't have enough energy to do this. consecutively back to back. I'll do 2 more probably later on in the evening, but I'm gonna take a nap for right now. Yeah, so I'm gonna go ahead and just kill it right here, and then we'll continue on later. All right. Toodaloo. Bye bye.